Nestled in the middle of the Himalayas is the Tibetan plateau, a large, flat, largely grassy expanse with an average elevation of over 500 meters. At such heights, the air is thin, and because of the surrounding mountains, the region receives little rain. It's a cold, harsh environment, one that many animals simply aren't cut out for. Homo sapiens managed to settle in this unforgiving landscape around 30,000 to 40,000 years ago, and around 10,000 years ago, they brought their dogs. While that might suggest our species as especially rugged or adaptable, we now know that neither the people nor their pets toughed it out alone. Both cribbed DNA notes from other species in order to adapt. Either before, during, or shortly after their migration to the plateau, H. sapiens got friendly with Denisovans, while their domesticated dogs interbred with Tibetan wolves. And from those hybridizations, both picked up adaptive variants of the EPAS1 gene, which encode version of the protein that help their bodies, and especially their blood, cope with lower levels of oxygen. You have the exact same phenomenon between dogs and wolves as you have between humans and Denisovans, explains Rasmus Nielsen, a geneticist with the University of California, Berkeley. It's so cool, but it turns out that, for the canines, that's not the whole story. Researchers refer to these extinct species, whose genes linger in the genomes of living animals, as ghost lineages, and we now know they're everywhere in the tree of life. They simply remained obscured until recently, when advances in sequencing technology and genomic analyses began to reveal them. Discovering when and where species of humans interbred with and interacted with each other will tell the hidden stories of our past and help us understand why H. sapiens is the only hominin species left alive today. Now, scientists are realizing those matings aren't just fruitful in the sense that they produce surviving offspring. I think there's a growing sense that this could be a way for a population or a species to quickly adapt as it moves into new environments, says Sankararaman. In fact, this kind of adaptation via hybridization, or what's often referred to as adaptive introgression, seems to happen all the time. The idea that hybridization plays a significant role in evolution is old hat to botanists, but fairly new for zoologists, from within the last 5 years, says Nielsen. The prevailing view, thanks to influential 20th-century biologists such as Ernst Mayr, had been that the commingling of distant relatives was rare and of little importance, especially in mammals. See, hybrid animals are not nature's misfits. One of the earliest pieces of evidence for adaptive introgression in mammals came from a 2015 study on domesticated pigs, Sus scrofa domesticus, where a ghost Sus lineage was uncovered. Researchers in China were looking for genomic signatures of adaptation to northern latitudes, and, therefore, genes that may confer cold tolerance, in 11 domesticated breeds when they spotted something strange, a 14 megabase region of the X chromosome that not only differed between northern and south. A 2015 study in Nature Genetics found that pig breeds from northern China, such as the Meishan pig above, possess DNA from an unknown, extinct Sus species. When the team created an evolutionary tree for that chunk of the genome, which included their domesticated pig breeds as well as Chinese and European wild boars, S. scrofa, and 4 other pigs, genus Sus, they found that the southern breeds clustered with the other Sus species, as one might expect. 2 years later, the same research group found another gene, this time, one that may have been involved in domestication, that also appears to have entered the domestic pig genome through hybridization with another, as yet unidentified Sus species. Whether people intentionally bred their pigs with other swine species or just happened to select for genes from a natural hybridization event is unknown. Either way, these findings are far from the only documented examples of adaptive introgression. In addition to the EPAS1 examples in humans and dogs, genetic research has confirmed that Western European house mice, Mus musculus domesticus, obtained a gene conferring resistance to the rodenticide warfarin from the Algerian mouse, Mus spretus, and Gulf killifish, Fundulus grandis, can tolerate H. These and numerous other instances of adaptive introgression from recent years have bolstered the idea that hybridization is a key mechanism for evolution. It's a new way of thinking about evolution, that really species. Are not isolated, they're connected to other species, says Nielsen, and when the environment changes, they can pick up DNA to adapt to new environmental conditions. See, Neanderthal DNA in modern human genomes is not silent. If that's broadly true, then looking for ghost sequences could be a He search for a genetic legacy of unknown species, particularly adaptive introgressed variants, in the genomes of extant livestock and crops will provide new sources of genetic variation for breeding and therefore help solve a pressing issue for humans, they write. Humans have long been trying to capture such traits through hybridization. Some 10,000 years ago, people bred Chinese pigs with European ones, passing along key fertility and immunity traits to the latter. Similarly, research has revealed that domesticated cattle in China were bred with yak and banteng, a species of cattle endemic to Southeast Asia, to help them survive high altitudes and tropical environments, respectively. Modern genetic science enables a more surgical approach, using gene editing to insert specific genes or variants into animals' genomes, rather than producing hybrid offspring with a mishmash of genes from different species and using selective breeding to fine-tune the traits of future generations. This type of gene editing is already being done with plants and is being explored in livestock, so in the future, ghost DNA could be targeted to confer desired traits to the plants and animals we cultivate. Windows on the past. Not all of these spectral sequences are adaptive. Still, even genomic ghosts that have persisted through chance could prove invaluable to researchers, as they may reveal novel insights about evolution and the ecology of bygone ecosystems. Studies on ancient introgression in felines have noted that interspecies dalliances have a marked impact on our ability to accurately reconstruct evolutionary relationships, so they're important for evolutionary biologists to consider when reconstructing the tree of life. The identified ancient genes suggest that the origin story of domesticated pigs is more complex than previously thought and point to gaps in our knowledge of the ecosystems where domestication occurred. After all, the genomic findings imply that there are two species of pigs we've never sequenced that were common enough in the past to leave a genetic footprint on our swine. Technical and other challenges. The investigation of genomic ghosts for ecological and evolutionary purposes is still in its infancy, as cutting-edge statistical methods for detecting ancient introgression have only recently been developed. Plus, these methods have mostly been designed to delve deeper into the hybridizations that occurred in hominins, says Martin Kuhlweim, an evolutionary biologist at the University of Vienna. Because of that, they may not work as well in other species. For instance, many of these methods require full genomes from the ancient relatives in question, something we have for Neanderthals and Denisovans, but which are rare for other extinct animals. Still, the field of ancient DNA is exploding, so it's not hard to imagine a future where scientists can employ tools developed for human ancestry studies on any animal species, yielding information that could help explain why they, and not their ghostkin, are the ones still around today. His work on chimpanzees and bonobos, Pan troglodytes and P. paniscus, our closest living relatives, is a perfect example. These animals are similar in many ways, but they differ markedly in behavior, with bonobos often noted for being less aggressive and more sexual than chimps, and have subtler variations in their physiology and ecology. The researchers were able to reconstruct an estimated 4.8% of its genome from their samples, but widespread sequencing of bonobos could reveal much more, allowing researchers to dig into questions about the ape's physiology, answers that could provide novel insights. Unfortunately, research into the ghosts and animal genomes is racing against the clock, says Kuhlweim, because it relies on sequencing many genomes from extant species. I think the main obstacle right now is the speed at which these species disappear. Finding enough genomes from wild individuals and getting them sequenced is becoming a challenge. According to a Science Advances paper published July 16 that Shapiro co-authored, about half of the human genome can contain sequences from introgression events, during which DNA flowed in by mating with Neanderthals, Denisovans, and potentially other as yet uncharacterized hominin species, even though it's probably in there, in that little, tiny portion of the genome where nobody has any archaic DNA. Where we really need to look hard for those genes that make us unique, she says. Please support my channel to grow by pressing subscribe button and the bell icon. We will notify you technological news. Thank you.